In this part of our study of the ear, we will deal with the mechanisms of equilibrium and hearing. The equilibrium sense is not easy to describe because it does not see, hear, or feel. What it does is respond to various head movements. The equilibrium receptors of the inner ear, collectively called the vestibular apparatus, can be divided into two functional arms. One responsible for monitoring static equilibrium, and the other involved with dynamic equilibrium. Within the membrane sacs of the vestibule are receptors called maculae. They are essential to our sense of static equilibrium. The maculae report on changes in the position of the head in space with respect to the pull of gravity when the body is not moving. Remember that. Because they provide information on which way is up or down, they help us keep our head erect. Each macula is a patch of receptor hair cells within their hats embedded in the otolithic hair membrane, which is like a jelly-like mass studded with these little otoliths. They're like tiny little stones made of calcium salts. As the head moves, the otoliths roll in response to changes in the pull of gravity. And this movement creates a pull on the gel, which then in turn slides like a greased plate over the hair cells, bending their hairs. These hairs then send impulses along the vestibular nerve to the cerebellum of the brain, telling it, where is my head? So now you know that we all have rocks in our heads. As you can see here, the otoliths float around, and when the head moves, it bends the hairs. And in this diagram, you can see that the hairs move in the direction that the head moves. The dynamic equilibrium receptors found in the semicircular canals respond to angular or rotary movements of the head rather than to straight line movements. The semicircular canals are oriented in three planes of space. So regardless of which plane one moves in, there will be receptors to detect that movement. Within the ampulla, which is a swollen region at the base of each semicircular canal, is a receptor region called a crista ampullaris. These consist of a tuft of hair cells covered with a gelatinous cap called the cupula. Here you can see the ampulla and the crista ampullaris inside. Here we can see how the direction of the body movement causes the endolymph to move in the opposite direction. Then the cupula will move, sending information off to the brain. When your head moves in an arc-like or angular direction, the cupula will stimulate the hair cells. The stimulated hair cells then send an impulse of the vestibular nerve to the cerebellum. Let's now talk about the mechanisms of hearing. Within the cochlear duct, the endolymph containing labyrinth of the cochlea is the spiral organ of Corti, which contains the hearing receptors or the hair cells. As the sound waves are transmitted by the ossicles from the eardrum to the oval window, it sets the fluids of the inner ear into motion. The receptor cells are stimulated when their hairs are bent or tweaked by the movement of the gel-like tectorial membrane. Once stimulated, the hair cells transmit impulses along the cochlear nerve to the auditory cortex in the temporal lobe. Here, we interpret sound and get hearing to occur. Wow. This is a cross-section view of one turn of the cochlea, showing the position of the spiral organ of Corti in the cochlear duct. The cavities of the duct labyrinth contain perilymph, and the cochlear duct contains endolymph. This is a detailed structure of the organ of Corti. The receptor cells, or the hair cells, rest on the basilar membrane. When the same sounds or tones keep reaching the ears, the auditory receptors tend to adapt or stop responding to those sounds, and we no longer are aware of them. This is why the drone of a running motor does not demand our attention after the first few seconds. This diagram shows that to excite the hair cells, sound wave vibrations must pass through air, membranes, bone, and fluid. This drawing shows what the cochlear would look like if it were uncoiled. Sound waves of low frequency that are below the level of hearing travel entirely around the cochlear duct without exciting the hair cells. But sounds of higher frequency result in pressure waves that penetrate through the cochlear duct and reach the scala tympana. This causes the basilar membrane to vibrate in response to certain frequencies of sound, stimulating particular hair cells and sensory neurons. In the bottom diagram, we can see that the length of the fibers spanning the basilar membrane tune specific regions to vibrate at specific frequencies. The higher tones are detected by shorter hair cells along the base of the basilar membrane, very similar to a pipe organ. Now, what do we need to know? Here are the key points. First of all, what is static equilibrium and what parts of the inner ear are associated with this type of equilibrium? What is dynamic equilibrium and what parts of the inner ear are associated with this type of equilibrium? And what is the mechanisms of hearing? All right, so here's the end of this particular show.